lot of great resources out there if you want to learn biochemistry or related topics. And so here is a walkthrough of some of my favorite resources.
So one of my biggest resources I have to offer is actually a page that has a linkified page with lots of different resources and links to how to access them and why I think they're helpful. And so let's just go through it. I've done a video like this in the past. This one's hopefully going to be a little shorter and it's going to be updated. I've added a lot of stuff, especially resources for things like computer programming as I'm learning. Okay, but let's go through these. Okay, let's start with the awkward part. My own resources. So I've tried to collate these like resources together in a way that are easy to help people. So for example, I have a lab techniques page where you can learn about a bunch of different experiments, all sorts of experiments. I have a glossary where you can find these different terms. If you search on my blog when you're on an article, then it'll highlight the terms that you can then actually click directly on those terms and get information about them, or you can just hover over them and the little like glossary icon will pop up. I apologize that my website's kind of slow, but hopefully it is still a helpful resource. So you can see here that some of the terms are underlined, and then if you click on it, you'll get the information, or if you just hover over it, you can get information about it, the definition and everything, as well as if you click on it, then you'll get taken to the glossary itself, the glossary entry. Okay, I also have a post on protein expression and purification, all of these different types of purification, things when it comes to molecular protein, to recombinant protein expression and purification. Speaking of proteins, I have a whole page on my blog devoted to them, all of these different proteins and all the amino acids. I also have a page on grad school and thesis things. People ask me a lot for advice about grad school interviews, applications, various things like that. And so I have all sorts of stuff that I've done on this in the past, as well as some of the helpful things I found along the way. A lot of those helpful things are databases, and so we'll go through some of them today. And I recently did a post on some of the ones that I find the most helpful, and the ones that really serve as like a jumping off point for getting more specific information about proteins or nucleic acids or chemicals. And speaking of that, I have a public Google Drive BV resources where you'll find different resources, including this databases for biochemistry, where I have a list of these databases and links. This is just like, you can download this. It's also, you can download it from the blog page on the article about the databases. So let's get to some of those databases, some other resources. So not just databases, but also things like books and websites I find helpful in various aspects of things. So just a heads up, so we'll be looking at various books and websites for general biochemistry content, as well as cool articles, some things that are really specific for doing lab work. So things like calculations and protein prediction, working with cell culture, DNA and RNA, things about like reading articles and making figures, as well as a lot on structural biology, so crystallography and cryo-EM, various resources that help you learn those techniques and use those techniques. Then also some learning computer programming, and then binding assays, because I did a ton of binding assays to test RNA protein binding during grad school. And so I had a bunch of resources that I found helpful for that. So obviously some of these resources are going to be more or less helpful for various people, but it's all there if you want to explore. So let's go through it. Okay, now that we've gotten this is my stuff out of the way, let's get on to some of the resources that I find really helpful personally. Although I do find my own stuff helpful, because sometimes, I can't keep all of that knowledge in my head all the time. And so when I forget things, I can go back and see what I post on it. So it's a helpful tool for me as well. Okay, so one of probably my all-time favorite book is Leninger's Principles of Biochemistry. It's on my bookshelves. And another great biochemistry textbook is Biochemistry: An Integrative Approach by John Tansey, which has a lot of cool connections to like real world things. Organic Chemistry, Organic Chemistry Makes Ridiculously Simple by Jean A. Davis is a really great book. Cell Biology by the Numbers. This is a really cool book by Rob Phillips and Rob Milo, and it does, there's also like a free online edition, and it will allow you to do kind of like back of the envelope calculations about various sizes and kind of rough estimations of how fast proteins do things and how many, I don't know, how much sugars in a cell. I don't know, various different calculations and really cool things that put things into concepts because like into context, because it can be really hard when you're thinking about these various molecules, like how concentrated are they actually and how big are they actually and how fast are they going. So this book is really helpful for that. Here's a free OChem book with a biochem angle. I haven't actually used it that much, but I learned about it. It's this Organic Chemistry with a Biological Emphasis by Tim Zuckerberg. So check that out if you're interested. Okay, for websites, Bio Numbers. This, speaking of like numbers and things, this is a really cool website. So this is just like they have this like cheat sheet type of thing, key numbers for cell biologists about the size of various cells and concentrations inside of cells and length scales inside of cells and genome sizes and error rates. So this is a really cool thing. And then this database, if you can search for all sorts of really all sorts of random questions you want, like how fast do you blink, or how many hairs are on your head, and various things like that. And so it's not loading right at the moment, but my internet's been being weird, so sorry about that. But it is really helpful. Okay. Lab Exchange. This is a cool site that I actually have some of my content posted on there, but this is not meant to be like a self-promotion. But they have a bunch of videos and interactives, so you can actually like interactively virtually pipette things. They have various graphics and text and all sorts of things from a bunch of different like collaborators, and so totally free resource, and really cool. Okay, the medical biochemistry page, there's some really cool information about kind of like the medical relevance of various enzymatic pathways and mutations in those pathways. It's really great getting into biochemical pathways. Okay, Chem Guide. This is really good for anything like chemistry. Speaking of chemistry, chemistry Luger texts. There's a lot of free open access text content. For physicsy things, hyperphysics, I often get here through Google when I'm trying to figure something out. AdGene. So you might know AdGene as a place that you order like clones through. So they're like a plasmid repository, and so they'll host plasmids that people have cloned so that other people can then use them. More on that in another post, but they have a lot really great educational resources through their AdGene blog. IUBMB, so the International Union of Biochemistry and Molecular Biology. So I am the training ambassador for them, and they're really, really awesome. And speaking of really, really awesome, they have a cool interactive version of this like metabolic maps, as well as downloadable versions of metabolic maps. One is on my wall right now. Okay, Explorins, speaking of IUBMB, this is their like official enzyme database with all the classifications. Here's more about the IUBMB if anybody is more interested. The EMBL EBI has a mechanism and catalytic site atlas, which will show you like stepwise mechanisms for enzymes, or at least the proposed mechanisms, because a lot of times it's not exactly known. Various handbooks, protein expression handbooks, baculovirus expression systems, as well. So yeah, I still have some of my content posted through here because I've done more extensive posts on some of these various resources as well. So here's a post that I did on these baculovirus, so you can express proteins in insect cells. Cytova, previously GE, has really great handbooks on protein chromatography for purification. And yeah, so here's a really great place to start, and they have good information about troubleshooting. I've also found they have really good like customer service and technical support. So thank you. I've had a lot of questions. Okay, for videos like full courses and types of things, Khan Academy is really good, as well as Bozeman Science. Tyler DeWitt has some cool chemistry ones with great examples. Some universities offer like free lecture series and course content. So UC Irvine Open and MIT OpenCourseWare, I've used a lot in the past. So these are just a couple that I found really helpful, like organic chemistry and introduction to chemical biology. Just some helpful articles. This Essential Chemistry for Biochemists that was published in 2017, super duper helpful. Okay, some resources for doing things in the lab. So for like designing and setting up experiments, open wetware protocols. It's like a wiki, so like Wikipedia, except not Wikipedia, but the same type of principle. And it has various different protocols, lots of really great ones, so that you can do it yourself versions of things like mini prep kits, various things like that you can make yourself. You can also just see what's in them. Close for Harbor protocols. This is a lot of protocols. Some are free for everybody, other than just you have to have your institution subscribe to them. Then Jove, this has a bunch of experimental protocols with like video demonstrations, and some of them are free and some of them your institution has to subscribe to again. The AAT BioQuest serial dilution calculator and planner helps you pan out site serial dilution. So when you're like diluting things to half and half and half and half and half, and like going down until you get really, really, really tiny amounts. Yeah, and here's more serial dilutions, but this tool will help you do it, calculate things for you, so you know like what 1 to 2, 1 to 4, like what should I start with, what do I want to end up with, there's things like that. Okay, dimensional analysis, like going between different units. These tools will do that all for you, but here's how you actually do it. Yeah, so Promega Biomath has some good conversions for like DNA and proteins. GraphMath molarity calculator. So molarity is this really key concept in biochemistry. We're talking about like copies of things, so like copies of proteins rather than just like mgs per mil, because some proteins are really big and some are really small. And so to give them like a fair comparison, we can use molarity because that counts the copies. Okay, Sigma has a bunch of calculators and apps. So does NEB. Here's some more like significant figures. It explains how to do that. So like how many things after your zero, like if you divide 1 by 3 and you get 0.333333, like a lot of times you don't actually have that sort of precision in your experiments. And so you have to know, okay, well, how many of those figures are actually like significant? And so more significant figures in this post that I did before. And here's a calculator, but it also helps you learn. Speaking of calculators, pH buffers. So this will help you know what to mix and how much molarity you want to whatever to make various pHs. Wolfram Alpha, I use it a lot in undergrad. They have some really helpful things for like math and physics, and they like help work out the examples and show you how they've done. ChemSpider has more information about any chemical. So there's some other ones that I have on my list of databases. Yeah, like PubChem is really great one too. Okay, for working with proteins, so I've done more detailed posts on these recently, and so there's videos and posts that are linked to us here. Uniprot, this is basically like your jumping off point for learning about a protein, and it connects you to a bunch of different databases. So it'll tell you about like the sequences of the protein, structures of the protein, functions of the protein, like various mutations that have been like found in the protein and whether there's anything known about them, like modifications to the protein. Basically, it has like a ton of different annotations for this protein, as well as links to take you to databases that have more information. So much more on that in a recent post that I did, as well as much more on XKZ prop param, which is a program where you can enter in a protein sequence or use an accession code that you get from Uniprot. So basically these unique identifiers for a protein. You can then use that to look up the protein. And if you use this XKZ prop param tool, then you can find out information about like the composition of the protein, how many of each amino acid does it have, What's the extinction coefficient? So you can go from UV to protein concentration, various things like that. The isoelectric point, the PI. This is going to tell you, like, is your protein going to be positively charged overall or negatively charged overall? And that's going to influence how you're going to go about trying to purify it. Okay, PFAM, this is information about protein families. Phosphosite Plus, if you want to learn about post-translational modifications like phosphorylations. CyPred, this will do things like predict the secondary structure, so things like helices and loops and strands and sheets, the various structures of a protein that are based on the interactions between the protein backbone as opposed to the side chain, so those like unique parts that stick off the protein. Predict protein, this is going to predict a bunch of things about a protein based on their sequence. There's also some online analysis tools, links. This page has a ton of links on, so more links than my page even probably. Biologics International has this like core codon usage table, and which is really helpful when you're trying to like figure out, okay, if I want to do like site-directed mutagenesis to make a change to the sequence, I want to make a change to the sequence that's not going to introduce a rare codon. I want to use one that the cells already use a lot. And so this codon usage table is really helpful for that. Okay, working with cell culture. So Thermo Fisher has this useful numbers for cell culture. This is I was told about recently, and it's really helpful. Letting me know like how much, like millions of seeds or cells, what does that correspond to? What kind of plating density do I want to have? This is really helpful for that. For working with DNA and RNA, sequence manipulation suite, this is a bunch of tools that you can do things like find the complement, the reverse complement, the inverse, all of these different terms and translations and various things, which can be helpful when you just kind of need to get the stuff into the format that you need it. WhatCut helps you find restriction enzyme cut sites. That can be helpful when you're doing cloning, as well as like introducing silent mutations that'll introduce a cut site so you can test to something there. AdGene I mentioned before. They have, as well as they have information about vectors. DNA SU, this is another plasmid repository, so like AdGene, typically it tends to have more than AdGene has, but often just cloned into like donor vectors. Basically like all of the sequences for all of like the human proteins and everything, they have like them all, but they might not be like formatted in some nice vector that's designed to be expressed or whatever. Whereas AdGene, they have a lot that are already cloned into vectors that you can use to express in like bacteria or insect cells or mammalian cells. So there's more like things like that, but there's more genes available on DNA as I found. NCBI BLAST, this will let you compare DNA sequences, like align them, find where they map to. So if you have a random sequence, maybe you were trying to do some cloning and you set it off to sequencing to see if your cloning worked, and you got back some random sequence, you're like, where the heck does this come from? You can then take that sequence, put it into BLAST and see where it actually came from. Maybe it was someone else in your labs. Okay, so BLAST is a lot of different things, as well as tools for primers. Here's some more about the different BLAST. They also have, like protein level things, so you can do things like search for protein sequences, do translated proteins, all sorts of different search tools, and so you can learn more about that, all of these things here. If you're interested in microRNA, this MirBase is a great microRNA database. Target Scan is good for finding the targets of those microRNAs. RNA fold web searcher will predict the secondary structures of single-stranded RNA or DNA sequences. So how they often think of them as just like these strands, but really they like fold up on themselves and do cool stuff. Okay, in terms of reading articles, more on this in blog posts and video I've done before. On Paywall, this is a browser extension that's basically going to put a little green lock on the side of your page, and then if something has a free version that you can access, and if it's gray, that means there's not a free version that you can access. But if it's green, you can click on it and it'll take you to a free version of it. There's also some like non SciPub, which lets you look at versions of articles often, but those are not always legal. So you might have that site blocked, various things like that. But Unpaywall just finds free versions that are legal. Okay. So I have, yeah, I thought, sorry, because I was thinking of PubPeer, which would help you read out bad papers. It's a site where people can comment on papers and people say, like, that figure looks like it was like reproduced from this part or whatever. But sometimes people put just like random other comments and they're not always bad. But this PubPeer, you can have this browser extension that'll put a little like flag on the page if it has a mention on PubPeer, then you can go and look at it. On site, this is a really helpful browser extension that generates this like sidebar that tells you the number of like positive, neutral, and negative or questioning citations. You can click on it and get a report of the snippets, like where something is cited. And so it's really helpful when you're trying to learn about the literature, maybe dive into a new field, and you want to know, okay, well, what came up, where did this idea come from, or where did this lead to? So this site is really helpful, as well as being able to see, okay, was this being cited because someone agreed, or like was this finding replicated, was it disputed, various things like that. When you're reading all these papers, you want to make sure that you're using a reference manager tool, so something like Mendeley or Zotero or EndNote or Pages. So you can learn more about these here, but basically you can see I have this like, I don't know if my little Zoom window is blocking it, but I have up here a Mendeley web importer that's going to allow me to, when I click on it, it will then import, I can have it import the article add it to my reference library. And then if I'm wanting to write a paper or something, I can then it'll insert the citation and I can find the paper later and various things. So reference managers are really, really key. Okay, for making figures, I use Adobe Illustrator. A free alternative is Inkscape. You want to use something that you can use that will make like vector graphics. So something like an SVG or PDF where you can blow them up really big and they're not going to get all like fuzzy, because what they're doing is they're actually telling the computer like, okay, make this figure by putting this point here, this point, like this here and this here and this here. Then you can like blow it up and it's not going to get like all pixely, unless you have like an actual like a picture in there. IBS, Illustrator for Biological Sequences. They let you make simple like domain architecture graphics. And then there's also like BioRender, which a lot of people use to make figures as well. It's a really great tool. Okay, I think there's like free versions and non-free versions. I think with the free version, they have like a watermark or something. I'm not exactly sure. Okay, structural biology. So if you're interested in protein structures, so we're talking things like finding out the shapes of protein using X-ray crystallography or cryo-EM. Speaking of those techniques, I have more posts on them in links to posts on them I've done before with a lot more information. The really place to start if you're interested in structures is the PDB 101. So the PDB is the Protein Data Bank. That's where you're going to find a bunch of different protein structures. And they also have a bunch of great like educational resources, as well as things involved with things like X-ray crystallography, cool resources for teachers and learners. They have like a Molecule of the Month series. They have free printouts that you can like fold up proteins, various cool things. SB Grids is like consortium that labs can join and that provides structural biology software. And they also have free lecture series on YouTube that often have talks by some of the software developers that are really helpful. They have like a really cool one on Chimera X, and it's like augmented reality features. So Chimera X is one of these, like, is a tool that you can use when you are like viewing protein structures and making pretty graphics of them. Another one is Pymol, and I've done various posts on them in the past. Okay, in terms of actually like for tools that I used in crystallography, and I was actually like solving the structures as opposed to just like looking at them. But when you're actually like trying to figure out, okay, so what is the raw data? How does that correspond to a structure? Some tools that are helpful with this are like Phoenix, Coot, Molprobity, which tell you some things about like, is that structure really accurate? Is it like, could those amino acids really be in that conformation, so in that shape? Is it geometrically too weird? Various things like that. 3D BioNotes, this is a cool site that lets you put in a PDB code for a structure, and then it integrates a bunch of information about it. Lets you see sites, mutations, etc. It's also good for a quick overview or look when you don't want to open up your PyMol. Crystallization constructs designer too. This is helpful if you want to crystallize a protein that's got floppy parts and you want to know where to cut off those floppy parts in order to try to get it to freeze in place for you so that you can use crystallography to get the structure of it. MPI Bioinformatics Toolkit also has a lot of tools for like structural prediction, homology finding. So if you're trying to find a protein that is similar to your protein, where there's a structure known of that other protein, and then you can like make a model of your protein based on the structure of that protein, or use that protein to help you figure out like your structure of your protein from your raw data, various things like that. Yeah, so like Swiss model is really good for homology modeling. Deep learning protein structure prediction tools. So like we're talking AlphaFold, Rosetta, Robetta, there's things like this. Books about crystallography, Introduction to Macromolecular Crystallography by Alex McPherson is really good. And I got to actually learn directly from him at Pilsner Harper Laboratories crystallography course, which is awesome. Another good one is Crystallography Made Crystal Clear, a guide for users of macromolecular models by Gail Rhodes. Which is a more meaty one for those interested in really getting down to those mathy details. Some articles, Protein Crystallography for Non-Crystallographers, or How to Get the Best but Not More from Published Macromolecular Structures by Vador et al. This is one of my favorite articles ever, super duper helpful. I've gone back to it over and over and over. It helps you understand things like R factor and B factor and all of these various things. Okay, so like, how do you know if the structure is good? Okay, some websites, resources, various different things, and lectures, all sorts of different things. Okay, so now this is the stuff that I've added more recently, things on like learning computer programming. I'm not talking about going like deep in, I'm talking about like getting started. So I'm not an expert on any of those things. But this is stuff that's I found helpful. So cheat sheets, some really, really great cheat sheets. Yeah, so some of these links don't seem to be active, but I'll make sure that I activate them on my website. Sorry about that. So cheat sheets for like R tidyverse. So I've done a post recently. I thought I had linked to it here, but I will add it. I recently did a post on like why it's important to learn these various tools and some of the basic tools that you should learn. So if you want, for scientists, the most helpful things are like learning R, Python, which are two different programming languages, and like some shell, so like some command line things. And so learning the basics of these is going to be really helpful. And I have a post that I will link to about why it is so helpful. But in terms of actually using them, there can be a learning curve. So for R, there's this thing called the tidyverse. You can learn more about the tidyverse here, but it kind of makes things more simple for you. If you're using R, you're going to work with R Studio, and so more on this in the other posts that I've done that I will link to. And our tidyverse is made by the R Studio people. Basically, R Studio is going to allow you to have like a sort of like graphical user interface, so even though you're typing in code, you can actually see things. When you're using like Python, so Python, you often want to work the Jupyter Notebook, which is kind of like this web-based browser that you can then work on your code in. More on this in the other posts, but it's really helpful. And so you want to check out Jupyter Notebook, and they have this, here's a cheat sheet for that. For when you're using Jupyter or Notebook, or even if you're not, when you're using Python, if you're wanting to do a number of type of things, you're probably going to be wanting to use NumPy and Pandas. So Pandas is kind of like Excel, except that you're doing everything in code and you have a lot more control. But it also offers a big learning curve. So here are some cheat sheets for it. For graphing, Matplotlib in Python, and ggplot2 in R. So here's some Matplotlib cheat sheets. For command lines, here's some Mac terminal commands cheat sheet, really good stuff. Okay, some good YouTube videos. I've been watching a lot of YouTube videos as I'm trying to figure things out. Here's Introduction to R, information on YouTube, Jupyter Notebook tutorials. introduction to the tidyverse. The RStudio has like a whole channel. Project Data Science has a bunch of cool videos on Python. Here's a cool one on pandas. This one is one I'm watching recently. It's like 5 hours long, but it's like 50 most popular Linux and terminal commands, which is super helpful because I found that like terminal is the thing I'm like most uncomfortable with. So here are just some other links. So I mentioned ggplot2. There's like a site where you can go and look at example graphs as well as the source code to make them. Various tutorials on various aspects of things, NumPy, BioPython, command line R, all sorts of stuff. And then from grad school, yes, that random stuff on people if you're doing binding assays. GraphPad Guide has some really great things, more on slot plots, which I did a lot. Just simple and informative binding assays. This is really, really helpful, as well as how to measure and evaluate binding affinities. These are really, really helpful if you want to do any sort of binding experiment. It's going to help you set it up correctly and interpret things correctly. Okay, so hope that was helpful, and I will add this video to this page as well. And hope that you found something in here that was like, oh, that'll be helpful for me. So yeah, so happy experimenting and doing whatever you're gonna do and learning, and just enjoy learning.